This video is a continuation of part 1 of Osmosis, where we're looking at the basic mechanism of osmosis. Here, we're going to look at tonicity and how you apply this to osmosis. Then we're going to talk about a number of applications of osmosis, as well as have several questions to check your understanding. When we're looking at tonicity, this is the measure of osmotic effect of two solutions separated by a cell membrane. It essentially describes the behavior of a cell when placed into a solution. It refers to the ability of a solution outside the cell to change the shape or tone, tonicity, of a cell by altering the cell's internal water environment. And we have three types of tonicity that we use in osmosis. We have what's called an isotonic solution, a hypotonic solution, and a hypertonic solution. In an isotonic solution, it has the same concentration as a cell. Outside of the cell and inside the cell, it's the same concentration. Therefore, water is going to move into and out of the cell at the same rate. There is no net movement of water. It's in a state of equilibrium. You want to mark these. Make sure you know that these are examples of isotonic intravenous solutions. Intravenous means within the vein. A 0.9% saline solution is isotonic to the cells. Lactated Ringer's solution is also isotonic. A 5% dextrose is initially isotonic, but then once the body metabolizes the dextrose, it can become hypotonic. And solutions with about 295 milliosmoles per liter of solutes. Well, the range is actually 275 to 295 milliosmoles per liter. And I'm going to go over this when we get to the concentration of body fluids. But for right now, just know it's about 295 milliosmoles per liter. Well, what about a hypotonic solution? Well, this has a lower solute concentration, thus a higher water concentration than inside the cell. Water's going to move into the cell by osmosis, causing it to swell and possibly burst, a process called lysis. What about a hypertonic solution? This has a higher solute concentration, thus lower water concentration than inside the cell, and water's going to move out of the cell, causing it to possibly shrink, which we call crenation. Let's test our knowledge. Here we have a red blood cell. These arrows are representing water moving into and out of the cell. What type of solution is this cell in? Isotonic environment. Here's another one. Are these red blood cells in an isotonic, hypertonic, or hypotonic environment? And what's the technical name given to the process these cells are undergoing? We see a larger arrow, water moving out than in, so these cells must be in a hypertonic environment, and we call this shrinkage of the cell crenation. Here's another example. Is this red blood cell in a hyper, iso, or hypotonic environment? And give the technical name for this process it's undergoing as it swells. Well, this cell's in a hypotonic environment, and the process is called lysis. So remember, when you're talking about tonicity, it always refers to the concentration of non-permeating particles of the solution outside the cell relative to the solution inside the cell. In the medical field, we're primarily only talking about the concentration of the solution outside the cell. Now, if you get into a general biology class, then you're going to have to distinguish between inside and outside. But for medical purposes, if you just focus on outside the cell and what the concentration is, you're golden. Let me ask you a question. Do you think pure water is hypertonic or hypotonic to the cell? It would be hypotonic because pure water only consists of H2O molecules. It would be like buying deionized water. It's only H2O. There's no solutes in there. So since there's no solutes, the cell has solutes inside of it, pure water would be hypotonic to the cell. And plants use osmosis in order to prop themselves up. When you look at a plant and it's wilted down, it needs more water. So you put water in it and then it will perk back up. Well, what exactly is happening? Plants have a cell membrane and they also have what's called a cell wall. And the cell wall is a rigid structure made of cellulose. When you water a plant, that water moves into the cell by osmosis, and the cell begins to swell. Well, as it swells, it pushes up against its cell wall, the rigid cell wall, which provides more rigidity to that cell wall, and then the plant perks right back up. We call this turgor pressure. And we can also use this clinically for humans. Signs of dehydration is if you pinch the skin and the skin stays up longer than normal, that is a sign of dehydration. Skin with decreased turgor remains elevated after being pulled up and released. This is one thing you can do with your patients just to make sure that they're not dehydrated. Feeling thirsty. Let's apply what we know about osmosis to other physiological principles. Your thirst centers are located in your hypothalamus. Hypothalamus is part of the brain. It's just above the pituitary gland, and the two structures, the pituitary gland and the hypothalamus, are connected by this infundibulum. It's a stalk-like structure that holds them together. This hypothalamus produces hormones that actually control the pituitary gland. Well, the pituitary gland has an anterior lobe and a posterior lobe. This posterior lobe releases hormones that are made by the hypothalamus. And the two hormones it releases, well, one of which we've gone over in an earlier video, we're talking about labor. Do you remember what the name of that hormone is? Oxytocin. Well, there's another hormone that it produces, and it's called antidiuretic hormone. So the hypothalamus produces ADH, or antidiuretic hormone, and gets released into the bloodstream when the body needs it. An antidiuretic, that must mean you urinate less, because if you had a diuretic, it would cause you to urinate more. When you look at the functions of the hypothalamus, besides making these two hormones, how does it know when to release ADH? Well, in the hypothalamus, you have what are called osmoreceptors. Osmolality is the measure of the concentration of a solution. In this case, the hypothalamus is measuring the concentration of blood. During dehydration, when you have decrease in blood volume, you're going to have an increase in plasma osmolality, because as you lose water, your solutes in your blood become more concentrated. The hypothalamus is going to use its osmoreceptors to detect that. The hypothalamus is going to send signals to the higher areas of your brain indicating that, hey, you're thirsty, you should be drinking water. The hypothalamus is also going to produce antidiuretic hormone, which is going to go down this neuron axon and then be released by the posterior pituitary gland into the blood. That antidiuretic hormone is going to go to the kidneys and cause the kidneys increase water retention. What's that going to do? Well, that's going to cause a negative feedback loop where once you've increased blood volume, once you've decreased plasma osmolality, then the hypothalamus won't release as much ADH. Oh my gosh, osmosis! It's fantastic! What about if you're feeling constipated? Maybe you might want to take an osmotic laxative, such as magnesium citrate. Citroma is a brand name of a type of magnesium citrate. Maybe you want to take a magnesium sulfate like Epsom salt. Or you want to take Marilax. This has polyethylene glycol in it. Or you want to take one that has lactulose or sorbitol. These are all examples of osmotic laxatives. Well, how do osmotic laxatives work? When you take these substances, what's in them doesn't get absorbed by your intestines, so it remains in the intestines. Well, what does that do? Well, that creates a hypertonic environment in the intestines because of these solutes that can't get absorbed. Water gets drawn from the cells lining the intestine into the intestines by osmosis, and that helps to cause the fecal matter to loosen up. What happens to the water in the cells when it gets moved into the intestines by osmosis? Well, then you're going to have water around the intestines, in the bloodstream, that's going to move into the cells lining the intestine. It's going to replace that water. Now, you could do it the old-fashioned way like this, where you have somebody put on a big old glove and take out that condensed fecal material that's causing the constipation. This isn't a human. Well, this is a human, but this isn't a human. This is the cow. I don't know if they're helping to relieve constipation, but it gets the point across where you have constipation, it's better to take a laxative like this than something like this. And these right here are showing different osmotic laxatives. Well, what about the two individuals I mentioned at the very beginning of the video who suffered from water intoxication? Well, these two individuals are Jennifer Strange and Matthew Carrington. And on January 12, 2007, a 28-year-old Californian woman, Jennifer Strange, and a mother of 3 children died from drinking too much water. There was a radio show called KDND, and it was out of Sacramento, and they were holding a contest where they brought in individuals, and they had to drink as much water as possible, and they were looking at how long they could stop from peeing. The contest was called Hold Your Wee for a Wee, and that was that video game Wii. When it first came out, everybody wanted it, and so they were having a contest to see who could win this. What happened was that she drank so much water quickly that it diluted her blood. It made, what kind of environment do you think? It was a hypotonic environment around her red blood cells and around her other cells as well. So that causes water to move into the cells and swell them, especially the neurons. It also decreased sodium concentration around neurons, which are extremely important in order to synapse potentials. So by doing this, it caused her to die. Chico State student Matthew Carrington, age 21, died February 2nd, 2005 after a night of hazing with a water overdose. So there's a container right there that he had to drink, and fraternities were going, oh, okay, well, we're not going to have him drink so much alcohol that it causes him to die, so we're going to go ahead and just have him drink a lot of water. Well, they didn't understand osmosis, because as he drank all of that water, it did the same thing as what happened with Jennifer Strange. It diluted the blood, caused a hypotonic environment. Death due to drinking too much water. What kind of environment does it create around the cells when you take in too much water too quickly? This is going to cause a hypotonic environment, and water is going to move into the cells, causing them to swell and possibly burst. It also disrupts the sodium balance. So let's test our knowledge. Part 1 of osmosis. Use the following terms to complete the sentences. The terms may be used more than once, or not at all. Here are the answers. Here's part 2. Go ahead and pause the video. Here are the answers. And here's part 3. Pause the video. Here are the answers. So this ends the video, and please let me know if you have any questions on osmosis. Extremely important topic, both physiologically and clinically.